Hello everyone. Welcome back to Life of Fusion, where knowledge meets excellence. We are now in the part 6 of our lecture series Learn from PYQs for CSIR-Life Sciences. So if you liked our video, do subscribe and share with your friends so everyone will get help from this. So all other 5 parts have been uploaded, is there in our channel. You can go to our channel. We have a playlist called Learn from PYQs and you can watch all the other parts. So let's start for the part 6 different question. The first question, receptor for which one of the following protein span the plasma membrane of target cells but does not contain intrinsic protein kinase activity? Okay, so they here asks that these are the some protein ABCD are different protein, means they are the ligand for their specific receptor. Here they ask that which of these ligand will bind to their specific receptor, and they asked about the receptor. Which of these receptor spans the plasma membrane, means that receptor should be a what? Transmembrane protein. Transmembrane protein means it spans the plasma membrane, and it have a cytosolic part, it have a intermembrane part, transmembrane part, and it also have a external outside part, okay, or ligand binding domain. Okay, so binding of which of the following ligand does not induce any phosphorylation in the ligand? It means that there should not be any kinase activity. If you see epidermal growth factor receptor is EGFR, insulin binds to insulin receptor, IGF-1 insulin growth factor IGF-1 bind to IGFR. All these 3 receptors, after binding with their ligands, they induce a tyrosine kinase. They have a tyrosine kinase domain, means the tyrosine residue in the cytosolic domain undergoes a phosphorylation, which further induce the signaling downstream. Okay, means what? These 3 have a kinase activity, but the growth hormone don't have any kinase activity. Binding of the growth hormone to its receptor does not induce kinase activity, means does not induce any phosphorylation. See this picture, binding of growth hormones to its receptors don't, because activation of the receptor binds to different protein but don't induce any phosphorylation. So the answer is option D. Next, PR protein play important role during plant pathogen interaction. Column X represents some of the PR family protein and column Y represents their main properties. So during plant when it counteracts with their pathogen, okay, it's undergo some different role. Okay, means like defensive activity. Okay, so for that there are different PR proteins are there. Those PR proteins have different properties during plant pathogen interaction. So some of the PR protein like 2, 5, 12 and 14, and the different roles are there or properties are there in column Y. So let's see what is the role of PR proteins. So in plants, a total of 17 PR proteins are available in plant which got activated during different types of pathogen interaction. Okay, so what are their properties? So we have option for column your PR2. PR2 activate beta-1,3 glucanase. So beta-1,3 glucanase properties having your PR2. Then PR5. PR5 is having thaumatin like activity. So this is 2. Then PR12. PR12 having defensive activity is PR12. PR14 PR14 is having lipid transfer protein. Okay, PR14 is a lipid transfer protein. This is 3. So A, IB, B, 2, C, 1 and D, 3. Option C is the correct answer. So next. So it seems like it's a genetic question. So a plant that produces disc-shaped fruit is crossed with another plant that produces long fruit. All the F1 plants have disc-shaped fruit. When the F1 were intercrossed, okay, F2 progeny were produced in the following ratio: 9/16 plants with disc-shaped fruit, 6/16 plant with spherical fruit, and 1/16 plant having long fruit. Okay, so as you can see, first that there are 3 different shapes of fruit are here. One is disc-shaped, one more is long shape, one is spherical shape. Okay, so when disc-shaped plant and spherical shaped plant, and long fruit plant, disc and long are crossed, those all F1 were disc, means disc is the dominant character and long is the recessive character. Once more see, when their F1 were intercrossed, okay, they got the fruit ratio 9:6:1. 9 disc-shaped, 6 spherical shaped and 1 long shaped. Means what? Long shaped is a homozygous recessive. Homozygous recessive. Okay, then from the option it seems like that the shape of the fruits are governed by 2 genes. Okay, means in gene interaction. Okay, that's why we got 9:6:1, not 9:6:6:1. If there is a single gene, or if the genes are not interacted, okay, means you will get 9:6:6:1. No gene interaction. Okay, no interaction means 9:6:6:1. But we got 9:6:1, means there is a interaction between genes. Both the genes produce some different character. So it means that these long shaped, we got only 1 out of 16, means it is homozygous recessive for both the gene. So we have 2 gene A and B. So we can say long is a homozygous recessive. Okay, so then if you see our disc and your spherical, so you can say the disc is dominant and spherical is the intermediate. So it means that the disc-shaped fruit comes from the effect of 2 genes, or the spherical shaped fruit can also come from or from only 1 gene. Then here your long shaped means no interaction, spherical means half interaction, disc means all interaction. Question is, which one of the following option gives correct genotype of spherical fruit obtained in F2? So you have to check what is the row where you will get spherical fruit in the F2 generation. And all genes are homozygous recessive. Okay, that time you will get long. And the F1 are heterozygous. Maybe, okay, you can say maybe they are heterozygous. So that's why you get what? 9 having both dominant, A and B are there. 1 all recessive, AABB, and 6 having a single gene having dominant. Means so for spherical fruit, for spherical shape, the contribution of a gene is either A or B. For disc shape, both A and B contributes to give disc shape. So no contribution means long shape. Any one contribution means your spherical. So which of this option? C, option C, capital A and small b. Here capital small a, capital B. So only one means what? Here dominant genes are the contributing gene. So only one allele, one gene that is present your capital A and capital B, or either only one, they will give spherical fruit. So option C is the correct answer. Next, so corded wings, ebony body color, and sepia eye are three recessive mutations that occurs in fruit flies. The loci for these mutations have been mapped and they are separated by the following hypothetical map distance. So Cu to E 20 centimorgan, E to AC 12 centimorgan. The interference between the gene is 0.4. Interference is 0.4. So the mutant CUESE fly was crossed with a homozygous wild type fly. The resulting F1 female were test crossed to produce 1800 progeny. What number of fly in each phenotype class is likely to obtain in the progeny of the test cross? Okay, it means you can see they just here the question, this part is just to make you more confused. Okay, so see here, I'll simplify the question for you. So this is the gene order, also they gave gene order and gene distance. Okay, so when they give the gene order and gene distance, so and they mention that the interference is 0.4, and below whatever they written here, this is to identify what? To identify the what? Gene map or your genetic mapping distance. Okay, or to check the interference and coincidence. Interference and coincidence. So first you have to check coincidence. So what is coincidence? So what is the coincidence of getting a double crossover product or getting a single crossover product? Means the coincidence of getting a recombinant. The coincidence of getting a recombinant offspring from between the cross between these flies. Okay, so you have this distance between this gene. So first you have to, because what is the interference now? Let me say about interference. Interference means if there is a cross in between one gene, okay, this cross interfere between the cross in the other gene, nearby gene. Means for example, if there is a cross between Cu and E, for example, if there is a crossing over between Cu and E, so this crossing over interfere with the crossing over between E and Se. Okay, that's why it's called interference. Means the chance of the production of double crossover product is very less. That's why when I used to explain you about genetic mapping question, I used to choose first, you have to choose that double crossover product. The lowest number of progeny, so the number of genotype is the double crossover product because of this interference. Because the crossing over in between two gene, okay, interfere with the crossing over or inhibit the crossing over in the adjacent gene. Okay, that's called interference. Okay, so now we have to find that what is the how much frequency. So now if these two genes, so what is the recombination? What is the recombination frequency? Or this you get the Cu distance between Cu and E is 20 centimorgan. So the 20 centimorgan comes from what? The 20 centimorgan means recombination frequency, means number of recombinant offspring produced between these two genes divided by total number of offspring into 100 gives you the recombination frequency, which we used as a map distance between these two genes. Means from this distance you can identify how is the frequency of the progeny will produce when you cross between the two genes. It means that, for example, if is 20 centimorgan, so the frequency between Cu and E will be 0.20, and the frequency between E and Se will be 0.12. This is the frequency because 12% of the progeny, when you cross between these two genes, they will give you recombinant offspring. 12% of recombinant offspring produced from the two genes. That's why the frequency will be 0.12. So now, if you cross these two to defend this fly with their homozygous wild type fly, and the total number of progeny is 1800. So if 1800 progeny produce, so now we have to find what is the expected progeny, expected value, because we have a map distance. So what is the expected progeny which will show a crossover between Cu and E or E and Se? From Cu to E, the frequency is 0.20. So out of 1800, 0.20 frequency, okay, will give you what? Will give you single crossover product between Cu and E. So what we will do? We have to do this frequency between these two genes, 0.20 into total number of progeny, 1800. 0.2 into 1800, you will get it will be around 360. So the single crossover between Cu and E, you will get 360 progeny. And if you do a single crossover product between, this is I'll write here, single crossover between Cu and E gene, then for single crossover product between E and Se, you get 0.12 into 1800. 0.12 into 1800, you get 216 progeny. Okay, so you can see. Then this is the single crossover between E and Se. So double what is the expected? See, these are the expected value. Expected single crossover product, expected single crossover between Cu and E, expected single crossover between E and Se, then expected double crossover product. Means frequency between of Cu and E 0.20 into frequency from E and Se 0.12 into total number of progeny. Okay, you will get 0.20 into 0.12 into 1800, around 43.2, around 43, you will get double crossover product. Is it clear? Now see the option. Option A, non-recombinant offspring 1250, single crossover between Cu and E 334. What you got? Cu and E 360. Okay, it's not the correct answer. B, non-recombinant 1181, single crossover between Cu and E is 360. Yes. Single crossover product between E and Se is 216. 360, 216. Then double crossover product is 43. We got 43 double crossover product. So what is the correct answer here? The correct answer is option B. Is it clear to all of you? If you have any doubt in the question or something like that, you can send us an email. Our email is provided in the description. So this question, a student added DMEM culture medium which was pink color to growing liver cells. So this liver cells grows in DMEM medium supplemented with phenol red. See, pink color is there, means it is supplemented with phenol red. Phenol red, it's there because to check the medium whether it is getting any acidic medium or basic medium, means the pH changes. If pH of phenol red changes, it will become yellow color. Okay, how pH changes? When you grow cells, the cells from the cell waste will come out to the medium. Okay, and the cell waste will reduce the pH. So reducing in pH change the color of the DMAM to yellow. So this indicated, so 3 days later the medium color was yellow. So this indicated that change in pH of the medium. Okay, mean acidic pH. Then the following statement were made to describe the role of Gibbs free energy. See this question, very simple question, and it comes in part C. You can get easily 4 mark. Okay, reaction can take place spontaneously if reaction A to B can take spontaneously if delta G is negative. Yes, delta G should be negative so that the reaction will proceed in a single direction. Reaction can take place spontaneously if delta G is positive. No. Delta G provides no information about the rate of reaction. Not like that. Delta G estimation provides the rate of a reaction. Yes, delta G provides the rate of a particular reaction. So A and D are the correct options. So A and D is option C is the correct answer. Okay. So thank you everyone. Have a good day. So if you like, do subscribe and hit the bell icon and share with your friends and do subscribe to our channel and do promote. Thank you everyone. Have a good day.